Our second speaker is Dr. Selma Aybek. Dr. Aybek is full professor of neurology at Freiburg University, Switzerland, and is chair of the Committee on Research of the American Neuropsychiatric Association. Please welcome Dr. Aybek.
Ladies and gentlemen, dear colleagues, I'm very pleased to present today recent data on vulnerability factors in functional neurological disorders, or FND in short. FND are defined as clinical syndromes of genuinely experienced neurological symptoms. The pattern of deficits are consistent with dysfunction of the nervous system and show variability between or within tasks, as illustrated in this video, where the upper panel shows a severe weakness with impossibility to lift the left leg or flex the ankle, when the lower panel shows a very light weakness. So this is what we refer to as motor discordance, and is a positive sign to make the diagnosis. FND used to be a time and cost-consuming process when it was seen as a diagnosis of exclusion, but we now have reliable positive clinical signs, such as the motor discordance, to make the diagnosis. As no biomarkers exist, a lot of stigmatization of patients came because the disorder was long ignored. And luckily, over the last decades, a new interest for FND arose, and two main lines of research emerged. The first one is focusing on how symptoms are produced at the brain level. And many neuroimaging and experimental behavioral tasks have demonstrated dysfunction of several networks, in particular, enhanced self-monitoring, misdirected attention, and impaired sense of agency, which is the sense that we are the agent of our own actions. So altogether, these findings point towards impaired complex sensory-motor integration underlying the symptoms production. Then a second line of research is focusing on why do certain individuals develop the disorder, and here again many studies showed dysfunction in networks, in particular during emotion processing, during recall of trauma, and in relation to childhood abuse or neglect. All these findings point towards an understanding that there are abnormalities in emotion and motion network interactions. So the research on why the symptoms arise focused also on biological stress, because indeed adverse life events in adulthood and childhood are more frequent in FND than control groups. And however, a recent meta-analysis showed that in a variable proportion of cases, no traumas were reported before the onset of FND, meaning that traumas are not necessary to the diagnosis of FND and are not causal, but are strong risk factors, calling for more research into this stress diathesis hypothesis, or how do vulnerability factors are influenced by stress. And research in biological stress focused on measuring cortisol and the functioning of the HPA axis. But so far, data in FND have been contradictory when measuring cortisol in the saliva. Again, calling for more research. And this is exactly what our lab is doing with a series of studies. And what we're trying to do is to bridge together those two lines of research of how and why symptoms of FND arise. So I'd like to present two studies today, and the first one was just recently published in Brain, and if you want more details about it, you can come to the poster presentation on Thursday. So this was a work done by Samantha Weber, a PhD student in my lab, now a postdoc. And the aim of the study was to look at the interplay between traumatic life event, cortisol, and brain volumes. And why volumes? because it has been shown that chronic exposure to stress dysregulates the HPA axis, leading to a habituation phenomenon with ultimately a lower cortisol output. And a second mechanism of chronic exposure to cortisol is a neurotoxic effect on certain brain areas that have high density of glucocorticoid receptors, in particular the amygdala and the hippocampus. So what we've done is we assessed trauma with this 29-item self-report questionnaire, assessing the severity of different traumas and the duration. We collected cortisol in saliva samples at 9 time points in order to calculate the cortisol awakening response. And we conducted a voxel-based morphometry with the amygdala and hippocampus as seed regions. So our cohort consisted of 86 FND patients and 76 controls, matched for age and sex. The first significant result was that patients had a flattened cortisol awakening response. You can see the gray line compared to control in green. And the second finding was that patients had higher severity and longer duration of emotional neglect. So we were then interested to see, are traumas here neglect and cortisol values, here the CAR, related? So in order to answer that, we conducted a multivariate analysis called partial least square correlation. And you can see in yellow the significant association. You can see the height of the bar represents the weight, so how much this factor contributed to the model. And then the direction of the bar indicates if it was a negative or a positive association. And we found a negative association between the CAR and symptoms and duration severity in patients only, the blue bars. Then a third significant finding was that patients had significantly lower volumes of the amygdala and the hippocampus than controls. So here again, we were interested to see, do these change in volume correlate to the cortisol values? And again, we conducted a PLS analysis. And we found a negative association between the volumes and the CAR, but surprisingly in controls only, the red bars, but not in patients. So there are two main conclusions that can be done with this study. So first, these finding of higher emotional neglect scores linked to a flattened cortisol awakening response confirms a role of psychological stress and a mechanism of habituation. And the second conclusion is that the fact that those reduced volumes were not associated with cortisol increase suggests that neurotoxicity is actually not the primary mechanism. And why is it important? Because it means we need to look for another mechanism. And possibly this volume reduction represents a predisposition to develop the disorder. Now, when we talk about predisposition, we can turn to genetic risk and or epigenetic changes in early life. And I'd like to show you some preliminary recent data on first epigenetics. So the very first study was conducted actually a couple years ago by our group, where we focused in a very small sample of 15 patients and 16 controls on the oxytocin receptor. And we found significantly higher percentage of methylation in patients compared to controls. Now, very recently, published this year, the group of Vera Spagnolo and colleagues from the group of Mark Hallett conducted a bigger study on a genome-wide methylation in a sample of 57 patients and 47 controls. They looked at common genes of stress, pain, and psychiatric disorders. And when looking at subgroups, in particular in participants who had a history of abuse and in female only, they found specific components that were associated to FND. So to summarize, it tells us that methylation has a role in FND, but we need to characterize more the specificity of these findings, because here it was mostly linked to biological sex and childhood abuse. Now, if we turn to genetic risks, well, the same group found in a single nucleotide polymorphism study a role for the serotonin pathway, because they identified that T carriers of the tryptophan hydroxylase had a higher age of onset of the disease. And currently, our group is also looking at genetic data on our cohort of 86 patients. And here, I'm just showing you preliminary data on the oxytocin. We find, again, this time not epigenetic, but allelic variation in this oxytocin with more AA carriers in FND compared to control groups, so possibly here a vulnerability factor. And we're particularly excited about this finding of the oxytocin because oxytocin is not only involved in the stress cascade, but also in early attachment processes, and has been recently shown to be very important in the precision weighting of predictive coding model that are important for the sense of agency and for different mechanisms underlying symptoms production in FND. Now, I'd like to turn to a second study, not on volumes, but on functional connectivity with the so-called dynamic lesion, with reference to Jean-Martin Charcot, who postulated a century ago that FND, then called hysteria or conversion disorder, was due to a dynamic brain lesion that one day we might be able to see. And actually, he said, once we'll have a powerful microscope, we'll see it. And we kind of have this microscope now in the form of advanced neuroimaging techniques, in particular resting state. And we conducted a 10-minute resting state analysis. We parcellated our data into 17 networks to obtain inter and intra connectivity matrix. We then compared both groups, and we saw significant differences in the connectivity between the salience and limbic network. So for the rest of the analysis, we focused on two hubs, the amygdala for the limbic network and the insula for the salience network. Now, instead of choosing a static connectivity analysis that assumes that the temporal correlation has an activity that is stable over time, we chose a dynamic approach that allows to see how the brain changes in state over the time course. And we selected the co-activation pattern method with this first step where we only select those images that have a high activity, so here the insula and the amygdala. And in a second step, we clustered the remaining images into patterns of co-activation that were looking the same. And once we've done that, there are different metrics that we can test. In particular, we can look at the duration, so how long does a participant stay in a brain state, or how often do they switch and enter a particular brain state. So here's what we found. With the seed as amygdala, we found three different patterns. For the insula, we also found three distinct patterns. And when comparing groups, we found that patients entered less often this particular state of co-deactivation of the default mode network with the amygdala, and also entered less often this pattern of activation, co-activation of the mesolimbic or dopaminergic network with the insula. So again, we were then interested to see how does it correlate with clinical values and cortisol. And in this other PLSC analysis, we found a negative correlation with symptom severity and a positive association with the CAR, meaning that patients who had more severe symptoms and these flattened CAR response were the one who entered those states less often. So in conclusion, what we can say about these different states on how and why these factors interact, we can interpret the pathophysiology of FND in a multifactorial model with different factors playing a key role. In particular, These reduced volumes of the amygdala and the hippocampus may represent biological vulnerability factors, but we need more characterization of the gene-environment interaction to understand them. Then psychological factors play an important role. In particular, neglect plays an important role, and the chronicity of trauma is important as it induces possible maladaptive habituation of the cortisol axis. And in turn, this is linked to abnormal switching between different brain states in patients, which seems to be linked to symptom severity and could potentially be important in how symptoms are triggered or maintained, and this is what we can observe as symptoms in our patients. And with this, I will conclude and thank my whole team in Switzerland for this work and you all for your attention.